Welcome to the deep dive. Throughout the pandemic, we've all watched this one single virus cause just the most, astonishing range of outcomes.
Absolutely. It's been baffling for many.
Right. On one street, you might have someone who barely notices it, maybe thinks they have a mild cold, and then literally across the street, their neighbor could be in the ICU fighting for their life.
Yeah.
This incredible clinical heterogeneity. I mean, this massive variation in how sick people get, or sadly, mortality rates, it's just far too wide to be explained simply by, age or general health alone.
Exactly. It really screams that the biggest variable isn't just about external factors like how much virus you are exposed to.
It points inward.
It points inward to the intricate biological wiring of the host itself. That's the crucial realization, and it really sets the stage for our mission today.
Okay.
We are diving deep into the genetic blueprint of susceptibility. Our focus is that complex, sometimes devastating interplay between our own host genetics and SARS-CoV-2 outcomes.
And specifically for certain groups.
Yes, specifically tailored to the findings that concern individuals of European ancestry. The core problem, the real challenge, is identifying exactly which specific prevalent genetic factors dictate not just susceptibility and severity, but maybe most troublingly, the post-infectious autoimmunity we're increasingly seeing.
So it sounds like we're really asking if there are specific genetic codes that sort of preload certain populations for a tougher fight against this particular virus.
It's a good way to put it.
So what are the main genetic systems acting as the gatekeepers and the referees in this whole conflict?
Well, we really need to investigate two fundamental systems. These govern how the virus gets in and then how the body actually responds.
Okay.
First, there's the Human Leukocyte Antigen system. You probably know it as HLA.
Right. Immunity cornerstone.
Exactly. It's the core of our adaptive immunity, the machinery that's responsible for presenting those little viral fragments to our T cells.
Got it. And the second?
The second is the Angiotensin-Converting Enzyme 2 receptor, ACE2.
Ah, the famous one.
The very same. It's literally the cellular doorknob that the virus turns to get inside our cells.
So, okay, on one hand, we have the immune system's presentation platform, the HLA, and on the other, the virus's entry point, ACE2. Now, our source material seems to suggest there's a kind of combined vulnerability in European populations.
Yeah.
Can you sort of outline the central findings? How did these factors intersect to create maybe a perfect storm?
Yeah, the research points pretty clearly to a two-part vulnerability that seems to amplify risk. The first part is essentially an immunological blind spot.
Blind spot.
Yeah. Certain HLA alleles, which happen to be quite common in people of European descent, they just seem to fail to mount an efficient initial T-cell response against SARS-CoV-2 peptides. They're kind of immunologically unfit for this specific job, you could say.
Okay, so that's part one. The immune response is weak.
Right. And the second part involves that ACE2 receptor we mentioned. There are variants of ACE2 that actually seem to enhance viral binding.
Make it easier for the virus.
Much easier, making cellular entry far too easy in those cases. And importantly, this dual vulnerability, it doesn't operate in a vacuum. It often piggybacks onto pre-existing genetic predispositions for autoimmune conditions.
Oh, wow.
So that turns a viral infection from, just an infection into a potential catalyst for severe disease and potentially long-term immunopathology.
Right, that connects a lot of dots.
Yeah.
Okay, let's dig into segment one then. The HLA system, the foundation of that T-cell immunity. We hear HLA thrown around a lot, but let's get specific. How do these molecules actually function, and why does their variety, their polymorphism, dictate whether someone, sails through or ends up with a severe case?
Okay, so you can almost visualize HLA molecules as the immune system's highly specialized reconnaissance unit. Their entire job is to patrol inside the cell, capture tiny fragments peptides of any foreign invader, in this case SARS-CoV-2, and display them prominently on the cell surface, like waving a flag. These displayed fragments are then examined by our T lymphocytes. Now, if the T cell recognizes that fragment as non-self and potentially dangerous, it kicks off a targeted immune attack.
Right. Standard procedure.
Yeah.
But the variety matters.
Hugely. The critical aspect is polymorphism. HLA genes are incredibly diverse. They vary drastically from person to person. It's one of the most variable parts of our genome.
So my HLA is different from yours.
Very likely, yes. Now, if your particular HLA molecule happens to bind a key SARS-CoV-2 peptide very tightly, very effectively, your T cells get a strong, clear signal. Attack now.
Okay.
But if your HLA molecule binds that same peptide poorly, or maybe not at all, Well, the T cells either get a weak, ambiguous signal or perhaps no meaningful signal whatsoever.
That's the blind spot you mentioned.
That is the immunological blind spot, and it's the absolute core of the T cell failure hypothesis.
So the hypothesis is basically saying that certain European alleles, maybe because of their shape or their chemical properties, are just bad at flagging these specific SARS-CoV-2 peptides.
That's the idea.
Which specific culprits did the sources identify? Which ones seem prone to this failure?
Well, several key associations emerged, particularly from studies in cohorts in northern Italy and also the UK.
Okay.
The sources pointed pretty directly to HLA-CO1 and HLA-B44.
CO1 and B44.
Right. Studies found these alleles were positively associated with the actual incidence of SARS-CoV-2 infection. The hypothesis is that they are permissive.
Permissive, meaning they sort of allow it?
Exactly. They actively allow the infection to establish itself more easily because they are simply unfit, biologically unfit, to trigger the necessary robust T-cell response against critical viral targets. They're like the molecular equivalent of a weak security guard, just letting things slip through.
That idea of permissiveness is really compelling. It suggests it's not always that the immune system is overreacting, at least initially.
Correct.
It's that the initial targeted defense mechanism is just subpar.
Precisely. And we also saw this pattern with what are called class 2 HLA molecules, specifically HLA-DRB115.01 and HLA-DQB106.02.
Okay, DRB115.01 and DQB106.02.
Yes. These were linked to higher susceptibility or higher severity in those Italian and UK populations we mentioned. Again, it suggests that the presentation of viral epitopes to helper T cells, which are crucial for coordinating the entire immune response, is somehow impaired.
So multiple points of potential failure in the system.
Definitely. But I think the finding that really demands the most attention involves HLA-DRB108.
DRB108. Why was that one so significant?
Well, this allele presented a really profound risk. It was associated with an almost doubled risk of COVID-19 infection in a very specific high-risk group: Italian solid organ transplant recipients and also candidates on the waiting list.
Wow, a doubled risk in a group that's already medically fragile, often immunosuppressed.
Yeah.
That magnification of risk is pretty sobering.
It really is. And what made this allele potentially so dangerous wasn't just the clinical observation.
No.
The clinical data was actually backed up by computational modeling, which provided the sort of mechanistic proof.
The how.
Exactly. The analysis showed that HLA-DRB108 alleles, particularly the dominant European subtype, which is HLA-DRB108.01, were generally unable to bind any of the tested SARS-CoV-2 peptides with high affinity.
Unable to bind any of them effectively.
That's what the modeling suggested. Think about the biological consequence of that. If the body's primary antigen presentation system for this specific allele essentially cannot recognize key parts of the virus,
Then the T-cell response is crippled from the start.
it's essentially paralyzed for those crucial epitopes. And this translates directly into a higher risk of infection and, critically, a significant correlation with mortality in COVID-19 positive patients within that specific cohort.
So they lack that essential defense mechanism needed to clear the virus efficiently.
Right, allowing uncontrolled viral replication to then drive severe disease.
It really sounds like carrying that specific allele could turn what might have been an early manageable infection into an existential threat, simply because the immune system is, as you said, trying to fight with one hand tied behind its back.
That's precisely the implication. And we shouldn't overlook other class I findings either. HLA-CO5.01 was also significantly associated with an increased risk of death in some studies.
CO5.01 too.
Yes. So when researchers look at these different associations, they don't just see random chance. They see a collection of specific genetic variants that collectively seem to fail at mounting a comprehensive and timely T-cell defense, specifically against SARS-CoV-2.
This picture gets even more complex, I imagine, when we move beyond just single alleles and start looking at haplotypes. The packages of genes we inherit together on a chromosome. Do these packages amplify the risk or maybe mitigate it?
Oh, they absolutely define the overall risk profile. It's not just about one gene, it's about the whole combination inherited together.
Right.
In those Italian studies, researchers identified quite contrasting genetic packages or haplotypes. There was a susceptibility haplotype defined by the specific combination HLA A01.01G, B08.01G, C07.01G, DRB103.01.
Okay, that's a specific combo.
A very specific combo. And it correlated strongly and positively with regional COVID-19 incidence and mortality. This really suggests that when these specific potential failure points, maybe ACE1, B08, CO7, DRB103, are all bundled together.
You get a major genetic vulnerability.
Exactly. It's like having a complete set of suboptimal security features all installed at once.
That makes sense. But you said contrasting haplotypes. Was there a protective one too?
Yes, conversely, a protective haplotype was also identified, HLA, A02.01G, B18.01G, C07.01G, DRB101104G.
A different package.
A completely different package. And this combination showed a clear negative correlation with incidence and mortality. It suggests that having this specific genetic complement actually conferred a significant degree of protection.
So that contrast really confirms it. In the European context, at least based on these studies, susceptibility isn't just about one weak link.
Not at all.
It's about the full configuration, the whole setup of the immune presentation system you happen to inherit.
Precisely. The entire haplotype matters.
Okay, that whole discussion really sets the stage beautifully for what happens after the virus manages to get inside. And this brings us neatly to segment 2. Here, we shift focus from that immune recognition failure to the actual mechanics of viral entry itself, the ACE2 receptor. We're moving from the T-cell battlefield, as it were, to the cellular front door.
Right. This is the very first barrier. While HLA dictates how well you fight the virus once it's inside and replicating, ACE2 determines how easily that replication can even begin. ACE2 is the host receptor, we know that now. And when we look closely at the genetic variations within the ACE2 coding region...
Changes in the gene itself.
Exactly. We find alterations that can profoundly change the binding affinity, how strongly it sticks to the viral spike protein, the S protein.
Okay.
And if that affinity is higher, well, the virus needs less effort or perhaps even a lower dose to initiate a critical infection.
It's like the difference between a key that sort of wiggles a bit in the lock versus one that just slides in perfectly effortlessly first time.
That's a great analogy. But let's use the specific language from the sources. Computational modeling was really crucial here.
The modeling again.
Yes. It detailed how certain genetic variations, which are particularly prevalent in European populations, fundamentally alter the energetics of viral entry.
Okay.
The most notable variant identified in these studies is ACE2 G211R.
G2LNR.
Right. This variant is found in approximately 0.2% of the European population. Now, that might sound small, but 0.2% translates to a significant number of people worldwide.
Sure does.
And this variant induced the largest calculated increase in binding energy compared to the standard or wild-type ACE2 receptor.
Okay, binding energy increase. That sounds like the core scientific finding here. The source gives a number 7.6 kilocalories per mole. Now, for those of us like me who haven't looked at biophysics since college,
Fair enough.
how do we contextualize that? 7.6 kilocalories. What does that energetic advantage actually translate to in terms of infection risk?
Well, it's massive. In molecular terms, binding energy dictates the stability and the strength of the interaction.
Right.
So if you imagine binding the virus to the receptor is like applying a piece of tape, maybe the wild-type receptor uses standard masking tape.
Okay.
An increase of 7.6 kilocalmol, that's like suddenly upgrading that tape to industrial-strength superglue.
Okay. Big difference.
Huge difference. It makes the binding approximately 7.6 kilocalmol more favorable, energetically speaking.
And practically, what barrier does that remove for the virus?
In practical terms, this energy change significantly reduces the barrier the virus needs to overcome to latch on effectively. It suggests that individuals carrying this G211R variant might require a substantially lower dose of SARS-CoV-2 viral particles to actually establish a successful infection.
Lower dose needed.
Yes, because the binding happens faster, it's stronger, and it's less likely to just fall off, to disassociate. This enhancement of binding efficiency essentially allows the infection to take hold more rapidly and more robustly than in individuals who don't have this specific variant.
So wow, even maybe a fleeting exposure or a lower dose exposure that someone else might fight off easily might be sufficient to establish a real foothold for these carriers.
That's the implication. And G2LONR isn't the only player here. We also identified ACE2 D206G.
D206G.
Yeah. This one is also most abundant in the European population, although at a slightly lower frequency, around 0.06%, still significant numbers. And this variant showed a strong increase in something called electrostatic attraction.
Electrostatic attraction. Why is that important for the virus getting in?
Well, electrostatic forces are basically the magnetic pull between molecules at a distance. They're based on positive and negative charges.
Okay.
The D206G variant seems to introduce changes that create a stronger initial charge attraction between the ACE2 receptor on our host cell and the spike protein on the virus. So, while G211R maybe increases the overall stability and energy of the final bond, the superglue effect,
Right.
D206G enhances the initial fishing net effect. It
Ah.
pulls the virus closer more efficiently, setting it up for that strong binding.
Okay, so the implication for European populations carrying these specific ACE2 variants seems pretty clear. A greater ease of initial infection.
That's what the modeling suggests.
And when you couple this potentially easier, more efficient entry, thanks to these ACE2 variants, with a potentially poor initial T-cell response due to those HLA variants we discussed,
Mm-hmm.
the high rates of severe disease we unfortunately observed start making, well, stark biological sense.
It really creates a kind of synergistic failure, doesn't it? The door is easier to open and the security system inside is compromised from the get-go.
Yeah.
That combination almost ensures the infection can reach a high viral load very rapidly, often before the adaptive immune system can even mount an effective targeted defense. And that leads us directly into the really dangerous territory of severe immunopathology.
That transition is perfect because it leads us straight into segment 3. The often devastating consequences of infection in these genetically susceptible individuals. We're talking about the autoimmune aftermath and the broader immune dysfunction. We're moving past that initial battle and looking at what feels like a resulting civil war within the body.
Yeah, and this stage is arguably the most frightening in many ways because it starts to explain not just acute severity drivers, but also potentially some of the lingering symptoms along COVID phenomena. We really have to look at how the immune system, maybe confused by a failing defense and perhaps that easy viral entry, starts turning on itself.
Okay, let's start with a concept called molecular mimicry, or MM. This is often cited as a key mechanism that might initiate post-viral autoimmune diseases. How does molecular mimicry connect back to those problematic HLA alleles we were just talking about?
Right, molecular mimicry is essentially immunological identity theft. It happens when SARS-CoV-2 shares molecular structures, tiny protein fragments called epitopes, with our own human proteins. They look similar at a molecular level.
Okay, accidental resemblance.
Exactly. Now, when the immune system trains a T cell to attack that specific viral epitope, if that epitope looks very similar to a self-protein, that T cell can become cross-reactive,
Anything.
meaning it starts attacking the similar-looking human protein in the host's own tissues. Mistaken identity leads to friendly fire.
And the connection to HLA, how does that fit in?
The connection to HLA is absolute. Remember, HLA molecules are the ones that dictate which viral fragments get presented to the T cells in the first place.
Right, the presentation platform.
So if a specific HLA allele one you inherited is particularly good at binding and presenting a SARS-CoV-2 peptide that happens to closely mimic a self-protein, well, that individual is genetically predisposed to an autoimmune reaction following the infection.
Wow. So these become dual risk alleles.
That's exactly the term used, dual risk. They increase risk on both fronts.
So the very same genes that might have made you more susceptible to getting the infection in the first place are now potentially predisposing you to developing conditions like, say, multiple sclerosis or lupus after the infection through this mimicry mechanism.
Yeah, it's the crux of it, yes.
Which specific overlaps were most critical in the sources you looked at?
Well, we have to highlight some specific overlaps that came up. Our initial culprits from the susceptibility discussion, HLA-C01 and HLA-B44.
The permissive ones.
Exactly, the ones permissive to infection. Well, they are already well established in the literature for conferring susceptibility to various inflammatory autoimmune diseases, even before COVID.
Okay, so that link was already known.
Yes. But perhaps the most striking dual risk involves HLA-DRB 108.01.
The one linked to doubled risk and mortality in transplant patients.
The very same. We know it's strongly linked to severe COVID-19 outcomes. However, this exact same allele also confers susceptibility to debilitating autoimmune diseases like systemic lupus erythematosus, SLE, and multiple sclerosis, MS.
Goodness. The clinical ramification there seems incredibly clear. A patient carrying DRB 108.01 who gets COVID-19 isn't just fighting the virus. They're simultaneously at a higher risk that the immune response to the spike protein itself could trigger the onset or maybe a flare-up of SLE or MS symptoms because of this molecular mimicry.
That is the profound concern, and again, the computational analysis reinforced this. It demonstrated that T-cell epitopes from the spike protein could indeed bind effectively to MHC alleles known to be associated with autoimmunity, including DRB 108.01.
So the mechanism is plausible.
Very plausible. The T-cells get trained on the viral threat, but the molecular resemblance is just so high that they proceed to attack, say, myelin basic protein in MS or nuclear antigens in SLE.
And you noted earlier that these kinds of autoimmune markers, or the HLA types linked to them, are particularly prevalent in this European population.
Yes, the sources emphasize that genetic markers associated with key autoimmune features like type 1 diabetes, for example, but also others tend to show their highest prevalence in Caucasians of European descent.
So that makes this intersection of COVID-19 and molecular mimicry a potentially heightened concern for this specific demographic.
It does. It might help explain, at least in part, the observed post-COVID rise in various inflammatory and autoimmune conditions that we've seen reported globally, but perhaps with some demographic skew.
Okay, moving on from mimicry, we need to discuss another extremely potent autoimmune mechanism the source has identified, and this is one that compromises the very first line of defense, the production of type interferon autoantibodies. This finding seems crucial for explaining some of the really high fatality rates we saw, especially early on.
Yes, this is a beautiful, if somewhat terrifying, example of how adaptive autoimmunity, the specific learned immune response, can actually create a catastrophic failure in the innate immune system.
It's adaptive breaking the innate.
Exactly. We need to remember that type I interferons, IFNs, specifically molecules like IFN alpha and IFN omega, are the core signaling molecules of our innate immunity. They are the absolute first responders.
The alarm system.
They are the alarm system. Crucial for shutting down viral replication immediately upon infection, even before T cells get involved, they tell neighboring cells to put up their defenses.
So if they're neutralized.
If they're neutralized, the virus gets a massive unchecked head start. And that's what happens with these autoantibodies. The body, through a genetically influenced process in the adaptive immune system, starts creating neutralizing autoantibodies, basically. Antibodies that attack and disable its own type I interferon.
So the body makes weapons that destroy its own alarm system.
Precisely. It's a catastrophic immunological failure.
How common was this? Was this autoantibody production strictly linked to severe COVID-19?
The link to severity was incredibly strong. These neutralizing IgG autoabs were detected in at least 10.2% of patients who are hospitalized with life-threatening COVID-19 pneumonia.
10% of the life-threatening cases.
At least 10%, right at the onset of their critical disease. And to really emphasize the link to severity, these autoabs were virtually absent in people who had asymptomatic or just mild SARS-CoV-2 infections.
Okay.
And they were also extremely rare in healthy control individuals sampled before the pandemic began in 2019.
So their presence is highly predictive of a really bad, life-threatening course.
Highly predictive, yes.
And the sources noted, I believe, that this phenomenon actually solved one of the really big mysteries of the pandemic, that striking demographic bias we saw towards severe outcomes.
That is probably the most striking takeaway from this line of research. The presence of these autoabs provided a powerful biological explanation for why we saw such severe sex and age biases in critical COVID-19.
Okay, what were the numbers?
The statistic is just compelling. Of 101 patients found to have these autoabs in one major study cohort, 95 of them were men.
95 out of 101, that's 94%.
A shocking 94%.
Yeah.
And furthermore, the prevalence of these autoabs dramatically increased with age.
So older men were vastly overrepresented.
Vastly. This condition, this production of autoabs against type I IFN, essentially acts as an acquired autoimmune funaki. It mimics rare inborn errors of IFN immunity. It severely compromises that crucial innate response, primarily in older men.
So that underlying genetic failure, where an individual's DNA codes for the propensity to create these self-neutralizing antibodies, translates directly into high vulnerability for the exact demographic that was most ravaged by the critical form of the disease.
It's the ultimate biological self-sabotage, really, and it demonstrates just how deeply host genetics controls the outcome, even right at the earliest innate phase of the infection. And importantly for our discussion, the cohorts used to detail this stunning finding were heavily centered on patients of European descent, making it central to understanding their disproportionate risk for these severe outcomes.
Okay, let's look now at the culmination of all this immune dysregulation in subsection C, the infamous cytokine storm. We know severe COVID-19 is often defined by this uncontrolled inflammatory response, which can tragically lead to multi-organ failure. How do our identified European genetic risk factors, the HLA types, potentially others, contribute to this hyper-inflammatory state?
Right. The cytokine storm is essentially the body's excessive damaging attempt to compensate for its failed initial defenses. It wasn't able to control the virus early, so now it throws everything it has at the problem, but in a chaotic way.
Okay.
It's characterized by an excessive production of pro-inflammatory cytokines, chemical messengers, most notably interleukin-6. IL-6.
IL-6, right. We heard a lot about that.
We did. And we can actually trace this tendency toward hyperinflammation back to the genetic background we've been discussing throughout this deep dive.
So are those HLA susceptibility alleles, we talked about the permissive ones, are they also linked to this inflammatory predisposition?
Absolutely. The very same alleles that seem to permit easier infection. Remember HLA-CR1 and HLA-B44?
Yes.
Well, they are widely recognized in immunology, even outside of COVID, for their established association with various inflammatory autoimmune diseases. So this suggests that carriers don't just have potentially a weak initial T-cell response to this virus. They might also have an inherent genetically defined tendency toward inappropriate, perhaps hyperreactive immunological reactions when things go wrong. They're sort of primed for inflammation.
Okay, and what were the clinical markers that signaled this level of immune dysfunction was happening in patients with severe respiratory failure?
Researchers noted signs of a profound systemic failure. One key marker was decreased HLA-DR expression on monocytes.
Okay, what does that mean?
Monocytes are crucial signaling cells in the immune system. When they reduce their expression of HLA-DR, another type of HLA molecule involved in presentation, it indicates they are either becoming exhausted or they're participating in a highly dysregulated inflammatory state. And importantly, this reduction was often linked directly to high circulating levels of IL-6.
So less presentation capability linked to high inflammation.
Exactly. It signaled a loss of proper immune control. We also saw severe lymphopenia.
Lymphopenia, low lymphocyte counts.
Yes, the depletion of critical immune cells like CD4 T cells, CD19 B cells, and natural killer cells. This is a classic hallmark of immune exhaustion and failure to control a high systemic viral load. The army is depleted.
So putting it all together, the body might be failing to recognize the virus effectively early on because of HLA issues, leading to high viral loads. Its own initial alarm system, the type I interferons, might be neutralized by autoantibodies in a significant minority.
Especially older men.
Right, and then the remaining parts of the immune system are both exhausted, shown by lymphopenia, and dangerously overreactive, pumping out IL-6, leading to the cytokine storm.
That really summarizes the devastating chain of events described in the sources. And, if we look just beyond the highly variable HLA system for a moment, there appears to be another common, widespread genetic factor contributing directly to those damaging IL-6 levels.
Oh, what's that?
It's a specific variation in the gene for IL-6 itself, the IL-6 174G allele.
A variant in the IL-6 gene. That sounds hugely important given IL-6 is the centerpiece of the cytokine storm.
It seems to be. This specific G allele, located in the promoter region of the IL-6 gene, is associated with increased production of the inflammatory IL-6 cytokine. People with the G allele tend to make more IL-6 when stimulated.
And is this common?
Its prevalence is significant. This allele is very common in European populations. Frequencies typically range from around 55% to 65%.
Wait, 55 to 65%. Hold on. That means the majority of individuals of European descent likely carry at least one copy of a genetic variant that predisposes them to a more excessive inflammatory response, like higher IL-6 production, should they develop severe COVID-19.
Precisely. This high prevalence points towards a widespread genetic background in this population that is, you could say, permissive to the very cytokine storm syndromes that unfortunately find so much of severe COVID-19 mortality. It really connects the dots between a common genetic tendency toward inflammation and the acute clinical severity observed disproportionately in some groups.
Wow. Okay, this deep dive has really revealed an incredibly intricate and, frankly, quite alarming interconnected chain of vulnerability. We've established that specific European genetics, certain HLA variants, certain ACE2 variants, appear to increase both the ease of viral entry and the likelihood of an initial T-cell response failure.
Correct.
And then, crucially, these same genetic backgrounds seem to drive potentially devastating immunopathology later on. This manifests as molecular mimicry leading towards autoimmunity, the dangerous creation of type I interferon autoantibodies that cripple the innate response.
Right.
And a generalized, widespread predisposition towards the cytokine storm, partly thanks to that common IL-6 gene variant.
It's a complex web, but the threads connect. The evidence presented in these sources is pretty conclusive. Outcomes during the pandemic were not just some random lottery. Susceptibility, the severity of the disease, and the subsequent risk of autoimmune dysfunction are profoundly and measurably influenced by the host's genetic background, particularly where these specific variants we've discussed are highly concentrated in the population.
So the heterogeneity of COVID-19 that seems so baffling,
Yes.
is in large part determined by the host's own genetic history, the instruction manual they were born with.
Exactly. It plays a major role.
So if this is the emerging picture of the genetic architecture of vulnerability, what does this deep knowledge actually imply for the future, say, for personalized medicine? What's the immediate actionable implication of knowing who carries these specific alleles?
Well, the sources definitely suggest a potential massive shift towards more proactive, more personalized health strategies in the future. Given the strong links that are emerging between these specific HLA risk alleles like DRB 108.01 and the propensity for autoimmune reactions after exposure to certain antigens,
Rather from the virus itself?
or potentially from synthetic antigens like those used in some vaccines. The recommendation that comes through in some of the analysis is quite clear. HLA typing in Caucasians could become essential.
Essential. Why is specifically highlighted as essential for this population in terms of risk mitigation?
Well, partly because a significant percentage of reported adverse reactions, particularly those potentially linked to autoimmune-like side effects following exposure to the spike protein, viral or synthetic, have been reported in individuals of European descent, where these HLA alleles are more common.
Okay.
So if a person is identified through HLA typing as being a carrier of, say, HLA DRB 108.01, that allele strongly associated with both severe COVID and MS and SLE susceptibility, that information becomes clinically invaluable.
Oh, so.
It moves us beyond just broad population-wide recommendations and towards a risk assessment that's tailored to their individual genetic instruction manual.
So this information wouldn't necessarily stop someone getting a vaccine or treatment, but it would maybe guide which treatments or prophylactic measures are considered best for them, or perhaps how those individuals are monitored afterwards.
Exactly that. Knowing the genotype allows clinicians to better assess and potentially mitigate the risk of that individual's genetic predisposition interacting unfavorably with specific viral antigens. It could allow for personalized monitoring, maybe watching specific autoimmune biomarkers more closely in those individuals after infection or vaccination. It might permit intervention before a full-blown autoimmune condition or a severe cytokine storm fully develops.
Right. Turning knowledge into prevention.
Precisely. This knowledge turns what seems like a fixed genetic vulnerability into an actual opportunity for precision prevention and personalized care.
That really is the ultimate takeaway, isn't it? The genetic architecture of susceptibility is becoming clearer, and it offers a potential path toward a future of personalized immunology that could genuinely redefine how we approach not just future pandemics, but maybe even common inflammatory and autoimmune diseases too. Thank you so much for guiding us through this incredibly crucial deep dive into the genetic battleground.
My pleasure. It's fascinating and vitally important research.